So hello, everybody. Welcome to today's CIC imaging lecture. And so today we'll be hearing a talk by Dr. Christopher Rowley, who is a postdoctoral researcher at the McConnell Brain Imaging Center at the Neuro at McGill University. He completed his BSc in medical physics and his PhD in neuroscience, both at McMaster University in Hamilton, Ontario. His doctoral work with Dr. Nicholas Bach centered around characterizing the cortical microstructure of the brain using MRI. His postdoctoral work in the labs of Dr. Christine Tardif and Bruce Pike has focused on developing more specific MR measures for brain myelination, with his most recent work on optimizing inhomogeneous magnetization transfer imaging. So if you have any questions, we can either take them throughout the talk or at the end of the talk. What would you prefer, Dr. Rowley?
It doesn't matter to me.
Okay, and you can also write them, so you can either write them in the chat or raise your hand.
Okay, so today I'm going to talk to you about using MRI to probe the microstructure of the cerebral cortex. So for this talk, I'm going to first motivate my work with an introduction into the relationship between structure and function in the brain. And then, because we have a group of diverse people, we're going to say why we want to use MRI to look at this. And then I'm going to briefly overview some of the work from my doctoral work that use qualitative metrics to study microstructure of the brain. And then I'm going to wrap up by discussing some of the work from my postdoc, looking at more specific metrics using physical and biophysical modeling for looking at the intracortical myelin content. So why study brain structure? I've taken this loop from a talk by Nicholas Weisskopf that I think really encompasses the importance of studying brain structure. So the loop highlights the connectedness of microstructure. I'm going to turn my laser pointer on. Function and behavior. So if we start at the most basic portion of the triangle, looking at function, or maybe in a disease case, dysfunction, we're looking at a basic input-output into the brain. So an input being maybe hearing something or feeling something, or output being able to move your leg. So if we start to group multiple functions together, we can get towards behaviors. So this might be carrying on a conversation or going for a run, playing sports. So if we have repeated functions turning into behaviors, we know from classical MRI, classical neuroscience with Hebbian wiring, that neurons that fire together wire together. So repeated behaviors end up changing the microstructure of the brain. There's also been recent work showing the myelination of the brain is activity dependent. So we have multiple ways that the brain structure is changing through repetitive firing of neural circuits. And then because we need certain structures in order to carry out certain functions, we ultimately have this microstructure having an impact on the absolute function of the region. So we see this sort of cyclical nature that encompasses plasticity and development in the brain. And this topic itself could eat up a few hours of time, but it's sufficient to say that MRI researchers are interested in developing tools to uncover the mechanisms that link these three. And I think to get a better appreciation of this, it's good to look at the scales that neuroscience occurs over. So if we look at the brain at the macro scale level, we're looking sort of on the scale of centimeters, and we might be looking to connect brain regions such as visual cortices and the frontal lobe. If with MRI, we get a little bit higher resolution, we get down towards millimeter scale, and we can start to see differences between brain regions. We can differentiate gray matter and white matter regions. So we get to see a little bit more anatomy in the brain. With histology, we get towards the mesoscale. So it's getting sub-millimeter resolution into the micrometers, and up top we have sort of a myelin stain where we see this nice striping and layer dependence of myelination across the cortex, where the darkest region sort of in the middle is the white matter and the outer region is the cortical gray matter. And below we have a Nissl stain for cell bodies, so all the dark purple areas are cell bodies. So again, we see a layer dependence of neuronal cell bodies in the cortical gray matter as well. So that's sort of some interesting structure that also relates back to the function. And if we go towards the upper end, we see the microscale where now we're down to nanometer resolution. So here, the best is really achieved with electron microscopy, but there's some super resolution light microscopy that's sort of coming out that's giving this another method to look at this, but that's sort of beyond this work. But really interesting with the microscale, we get to see the cell structures such as the lamellar wrappings of the myelin sheath around an axon. So in the span, we can see there's 7 orders of magnitude difference between the macro scale and the microscale. So MRI is realistically limited by physical constraints, especially for in vivo work. We're sort of limited to millimeters, and with higher field strengths, micrometers. But we can use biophysical modeling to attempt to extract some information that exists at the subvoxel level. So in the quest to correlate structure with function, it'd be best if we could probe the structure of the specimen while it's still functional. So MRI provides an excellent tool to do so, with many contrast mechanisms at our disposal to uncover the structure and composition of the brain. So a quick imaging modality comparison. So if you break your arm, you might go for an X-ray, maybe CT with some head trauma, or our favorite in our lab is MRI. So MRI provides superior soft tissue contrast because the signal we're getting is actually coming from the water protons, at least in the imaging that we're doing. And this is in comparison to X-ray, where the contrast you're getting is the attenuation of X-rays, and as well as CT. So we can see we get superior soft tissue contrast here. An additional benefit with MRI is we can generate contrast in a few different ways. I'm going to give a brief overview of 2 mechanisms that pertain to the work that I'll share today, but note there's several others that are used. So widely used contrast mechanism in MRI relates to the time that it takes for the net magnetization, M0, to realign with the main magnetic field, which is B0 here. The rate at which it does so is R1, and often known as 1 over T1 or vice versa. So this rate is strongly influenced by the main strength, or the strength of the main magnetic field, but also by the local magnetic field, which can be impacted by macromolecular and iron content. So this fact itself provides some local tissue contrast. So you can acquire multiple images, such as here I've drawn 5 lines. If you acquire different points on this time curve, you can fit this R1 metric. Alternatively, you can choose your sequence parameters and just acquire 1 image and get something with strong T1 or R1 contrast. So if we wanted to do a quantitative R1 mapping, we've acquired multiple images on this time curve. You can get a map that sort of looks like this. So this is an axial slice where we're looking from sort of the back of the brain to the front where the eyes would be up here. R1 maps are typically characterized by really strong gray matter, white matter contrast, where the white matter appears brighter than the gray matter. And because we're interested in the cortex, we can take these values and sample them onto the cortical surface and start to see regions across the brain differing from each other. So we see brightening of the primary motor and sensory cortices from the association regions in the frontal cortex. So another contrast mechanism that my work is interested in is magnetization transfer. So there's a few ways to generate this kind of contrast, but in my work, we apply an off-resonance RF pulse to the bound proton pool. So this provides selective saturation of the bound protons, and this magnetization exchanges rapidly with the water pool. So this change can be seen by imaging the water pool directly after the saturation pool, and we basically get an indirect measure of the effects of this pulse by seeing a decrease in this signal from the free water pool that happens from the exchange between the two. So if we capture an additional image that does not contain this sort of saturation RF pulse, we can begin to quantify this effect. And if we do that, we might get an image like this. So this one is specifically is an MT sat map, characterizing the saturation that occurs as a % signal drop of the image from the saturation pulses. Again, it looks very similar to the R1 maps, and we can also project this onto the cortical surface. So what are we seeing with this? So we turn to histological correlations with MRI signal to gain insights into the major contributing factors into the MRI signal and contrast. So some nice initial work was done by Bach et al. In 2009 in a marmoset that showed very strong spatial correlation between This is a silver myelin stain and T1 contrast. So the T1 contrast is, or R1 contrast, we have an inverse relationship between the two, but mainly we're seeing the same structural features come out. So V1, we have the Trajectories, and it shows up strongly in both. Another work that's highly referenced by Stuber et al. In 2014 also incorporated an iron stain, which tends to contribute quite highly to both T1 and T2 star contrast. They also incorporated a lipid stain for myelin content. And what this study found was that T1 or R1 contrast can be explained with a linear model, where most of the contrast is explained by the lipid content with some contribution from iron, where T2 star contrast is sort of flipped. It's predominantly described by the iron content and partially by lipids. And it's potentially most evident visually by looking at the Trajectories, which is very evident in the iron map here, very evident in the T2 star map, and a little bit less in both the lipids and T1 maps. So from a biological perspective, why bother looking at the MRI signal if it's going to tell us about myelin? What's so great about myelin in the brain? So while neurons are responsible for the computational processes that occur in the brain, they don't do it alone. The gray matter, which contains both neurons and glial cells, sees neurons being outnumbered by a factor of about 1.5. And if you look at the white matter, which is simplistically thought of as neural axons without neuron cell bodies and containing myelin as well, the ratio of non-neural cells to neurons exceeds about a ratio of 15. So given the abundance of these cells in a space that's really limited in real estate and being encapsulated by the skull, it must play an important role to justify both their numbers and the space that they're taking up. So the literature on the importance of the brain is rapidly growing. It's long been established that myelin plays an important role at speeding the conduction of neural signals across an axon, where we can see speeds exceeding 10 times as fast. More recently, emerging research field is showing that myelin is providing metabolic support for neurons, and this is also being aided by the fact that we're finding that myelination is an activity-driven process. So this is functionally changing where myelin patterns were thought to be quite static. So we now know that this is actually a very plastic process in the brain. And if we bring this back with the cycle I highlighted at the beginning of the presentation, we might be interested to know that myelin follows a regionally distinct pattern across the brain. So many people have used this patterning to parcellate the brain into small regions, where the hypothesis is that each of these small subsections of the brain is responsible for a specific processing task. So this again brings back the connection between the structure and function of the brain. So now that I've made a case for why we're interested in myelin and brain structure and how we can study it with MRI in vivo, I'll share some of my PhD work to start off. So again, with this work, we looked at qualitative MRI imaging, and the two applications I'll share today were in healthy aging and in bipolar disorder. So it's well established that the cortical gray matter in humans continues to develop well into adulthood. An initial study in 2015 with over 1,500 subjects, so that's quite a few, used the T1 divided by T2 weighted ratio to show that intracortical signal, which they're suggesting is reflective of myelin content in the brain, continues from roughly 17 to 35 in a linear fashion. A study that was actually completed 2 years earlier with slightly less subjects, almost 340, again using the T1 divided by T2 weighted ratio, looked at the age span from about 15 to past 80. And they found a nice inverted U trajectory across the age range, where at peak roughly, so these are just 4 sample ROIs taken, but it was around 50 years old. So 1,500, almost 350 subjects, that's quite a few subjects to look at if we wanted to look at clinical populations. So what we were interested in doing is if we could optimize the imaging, could we pull these trajectories out with the much fewer subjects? So we used sequence optimizations that were done by Bach in 2013 for T1 weighted imaging, because we know T1 is highly dependent on the myelin content in the brain while reducing our sensitivity to iron content. We also use the ratio approach similar to the T1 divided by T2 weighted, which helps remove a large degree of hardware-induced inhomogeneity in images. So if we see what this looks like, we have our high contrast T1 weighted image on the left. Instead of a T2 weighted image, we took a proton density weighted image, and you can really see the central darkening of this image, which is mainly receive gain differences, but we see that cancellation really comes out in the final ratio image, where it's a much more homogeneous image between front, back, and center of the brain. So we took this signal from the ratio in 67 individuals aged 17 to 45, and we sampled it across 3 cortical depths, where the 1/4 depth was closest to the pile surface, or CSF, and then moving inwards towards the white matter, we had the half depth and then 3/4 depth. So if we plotted that across the brain, again, pulling out just 4 sample regions, we see nice separation actually between the 3 cortical depths. And we're also finding this inverted U trajectory. So to simplistically fit this, we use the quadratic. You can look at the paper, we got the same results if we fit it with a spline as well. But with the quadratic, it's fairly easy to just pull out the age of the peak and the height as well. So this term H is the height or the age at the peak, and then K is the max signal achieved from this fit trajectory. So if we look at those values across the brain, the K value is fit vertex-wise, we see what we would expect, increasing myelin content as we move from the cerebral spinal fluid towards the white matter. Interestingly, this H term, which was fit on an ROI basis, wasn't statistically different between the cortical layers. So this is suggesting that the cortical layers are maturing at a similar rate. So the next thing we wanted to do was input these H values into a clustering algorithm to see if we could reveal development patterns across the brain. So the results from that were quite interesting. We do see a spatial distribution to this clustering, where visual and visual cortices were peaking earliest at around 34 years of age. Then we have association regions in the temporal and parietal lobes peaking next. And then in the frontal cortex, we sort of have this rostral, so sort of frontal to caudal towards motor cortex development, which matches a known functional gradient in this region of the brain. So it's worth mentioning here as well that this isn't simply tracking absolute myelin content. This is most obvious by looking at the comparison with motor and visual cortices, where the visual cortices are peaking earliest at about 34 and motor cortices are peaking somewhat latest, closer to 40. And these are two very highly myelinated regions in the brain. So in this work, we show that T1 weighted intracortical signal peaks between 35 and 40, depending on the region. This change over this time could be a compensatory mechanism, as we're not really expecting large cognitive changes during this period. And it matches other histological work showing elevated myelin levels in adulthood. So as was shown with this myelin stain by Kemper in 1994, and this was a myelin basic protein extraction from Su et al. 2015, also showing a peak in mid-adulthood. So we found that the development is consistent across the cortical depths, but this point itself should be revisited with higher resolution imaging that could be more sensitive to changes across the cortical depths. So the one other thing worth noting is the peak that we found was 35 to 40 is a little bit less than what I said from the previous work at about 50. So one thing that's also going on is, again, I mentioned we have reduced sensitivity to iron content. We know from MRI work such as T2 imaging and other histology that the brain is really good at taking on iron, but not so much getting rid of it. So iron accumulates in the brain with over age. So because we're not getting this contribution, I think we're getting a little bit lower of a peak compared to the T1 divided by T2 weighted ratio. Ultimately, there's a gap in the literature as to the exact changes that are occurring with the myelin sheaths during this age range. But given the recent optogenetic work in mice showing the myelin sheaths are highly stable in size, it's likely reporting changes in myelin registering as increases in new sheaths along the axons. So continuing on, With the imaging protocol validated in healthy controls, we collaborated with a psychiatric group at McMaster University to investigate microstructural trajectories in bipolar disorder. So a quick overview on bipolar disorder. It's characterized by cyclical episodes of depression and mania and affects about 1 to 3% of the population. So the onset is typically in the late teens to early 20s, which is why we were really interested in characterizing that age range from about 17 to 45. And there's also some arguments being made within the bipolar disorder research community for it to be considered as a developmental disorder. So this made bipolar disorder an interesting population to investigate with this methodology. Additionally, there's been myelin deficits reported in bipolar disorder with previous reports of decreases in cortical oligodendrocytes, which produce the myelin sheaths in the brain, and in white matter volume. So this is shown here in Uranova with a significant decrease between healthy controls and a prefrontal cortex region in oligodendrocytes, as well as this work showing decreases in glial cells combined. So if we added 40 subjects, which included a bipolar data set, again, age 17 to 45. And if we plot the age trajectories, we sort of see something like this. So these are 4 ROIs chosen because they come up quite frequently in the bipolar disorder literature. And the red line was what I showed you previously for the mid-cortical depth, but the blue line is now the bipolar disorder data. So what we really saw with this data set was a flattening of this trajectory compared to healthy controls. So this sort of potential compensatory mechanism that's occurring with healthy aging seems to not be occurring in bipolar disorder type 1. So to better quantify and visualize the flattening of that trajectory, we fit each group with a general linear model, where we're really interested in just the coefficient of this age squared term, which really describes the curvature of that trajectory. So if we look at the controls, again, we know that's in a strong inverted U, and we have a very negative coefficient. So that describes this inverted U shape. As we look towards bipolar disorder, this term trends towards 0, which indicates a more flat line. And this seems to be a global difference between the controls and bipolar subjects studied. And if we look at the significance of this coefficient across the brain, we see most significant regions ended up being the regions that peaked later. So again, these motor cortices regions, which are peaking potentially close to 40 years of age. So again, structure changes. We're really only concerned with that if it's also impacting the function. So what we did here was we looked at a score of verbal memory and how that was related to the T1 weighted contrast, which we're suggesting is related to the intracortical myelin level. So if we look at this term, we're not seeing, so these were, the colored regions are the significant regions that come up from the linear model. And there was no correlation between verbal memory and T1 scores in healthy controls. So this sort of comes back to it being a compensatory mechanism. The changes that we're seeing in the brain aren't resulting in a change in verbal memory. But in bipolar disorder, where there's pretty consistent decreases in verbal memory scores, we're seeing a correlation between the myelin content as indicated by this T1 weighted metric and verbal memory scores. So this might suggest that the people that are most impacted and not getting the increase in myelin that they should be getting with this age trajectory are faring worse on verbal memory scores. So the conclusions from this work is the development trajectories appear to be altered in bipolar disorder type 1. And this loss of signal correlates with verbal memory scores. So this work's still being conducted at McMaster University, but it's now being pushed through a longitudinal study to better provide evidence for these findings. So to summarize sort of my doctoral work and a little bit of notes on qualitative MRI, why we might want to do it. So qualitative MRI over quantitative is potentially shorter scan times, which is great if you're looking at clinical populations. It can be optimized to produce very high SNR and contrast to noise ratio in the images, which is always a good thing. Also very good especially if you're looking at or very interested in potentially rare diseases or disorders. There's often existing databases that exist where the images are available already. There's some cons to it as well. It's not all winning. It's difficult to standardize the values across sites due to changes in hardware, differences in hardware between the sites. And we have a loss of microstructural specificity. Easy example was the T1 divided by T2 weighted, where we lose that myelin specificity because of the increased iron sensitivity with the T2 contrast. So if we move towards my postdoctoral work, I started to focus more on various ways we can improve this imaging to get a more specific metric or measurement of the underlying biological structure. So one thing that's come up with most of my work at the Neuro is actually been looking at B1 inhomogeneity in the image. So the B1 field is the transmit field, and it varies spatially across the brain. So if we look at a B1 map, so this is a relative value where 1 would be if you wanted to apply a 10-degree flip angle. Everywhere with 1, that region of the brain is going to have that 10-degree flip angle. As we get out towards part of the cortex, we're seeing down to 80%. So we might only be getting an 8-degree flip angle. So this is going to affect our image contrast and our SNR values. So we can see if you're looking at the white matter, there's changes, but it's pretty close to 1. Because I'm interested in the cortex, it actually can drop quite a bit. And this will vary between different hardware. So if you go from one scanner to another, it'll be different. Additionally, this changes with B0. Also at the Neuro, we have the 7T scanner, so it's much worse at 7T. And to show you what that looks like, we acquired data in one person, image at both 3T and 7T. So I rescaled the 3T B1 map to be the same as the 7T to really see the difference. So we have the same upper end at 1.2, so 120% of the relative value. And that's mainly fixed at the upper end just for safety concerns. And then we really end up seeing the variation as a big decrease in signal. So again, the lower end was maybe 75% at 3T. At 7T, especially as you get down lower in the brain, if we're just looking at STX or single transmit, we're getting towards 30% or less. So that's a very large drop off. Again, if we were hoping to apply a 10-degree flip angle, now we're down to a 3 degrees. So that's quite a large drop. And again, this variation is quite massive over the cortex. So it's something that's a big concern. So again, I mentioned it affects contrast. So what does that actually look like in the brain? So one sequence that's often used is the MP2 range because it's pretty insensitive to B1 inhomogeneity, except it has its limits. So if we look at, again, as the same person imaged at 3T and 7T, we do have some received gain issues still at this 3T data set, but we won't worry about that. It's pretty easy on the Siemens scanner to deal with that. But what we are interested in is strong gray matter, white matter contrast, which we see throughout the image at 3T, especially in the temporal lobe and the cerebellum. At 7T, really where I indicated with this arrow is in the temporal cortex and even in the cerebellum, it really just, you lose the contrast between gray matter and white matter. So if we look at the B1 field, what's going on here? So I rescaled this again because we're really getting a drop off in the B1 field. So now we're all the way down to 0. If you use a cursor and look at what the B1 value is here, we're actually getting close to 20, 10%. So because of that, we're not able to get the signal difference between the gray matter and white matter contrast with such a low flip angle. So I've made a case for why this B1 correction is important, whether you're doing quantitative imaging or even if you're doing standard anatomical imaging, you're not going to be able to segment this image because there's no way to tell the difference. So for me, I'm interested in quantitative imaging. So we're looking at correcting our quantitative maps for this B1 inhomogeneity. So going back, we know that R1 longitudinal relaxation rate is quantified by this equation. So to correct for B1 plus, It's relatively straightforward. You can use your B1 plus map that you acquire additionally as a scaling factor on your excitation pulses. So this can either be used in signal equations such as this, or in block simulations if you have a more complex T1 mapping or R1 mapping approach. So using the same MP2 range at 3T, I'll show you what that might look like. So for MP2 range R1, so this is a 25 subject average. We have a B1 map that's looks like this. Pretty reasonable left-right asymmetry, but we have sort of this darkening region in this medial prefrontal cortex. If we look at uncorrected value to the untrained eye, it might look pretty good. We have, again, good left-right asymmetry. The motor sensory cortices are showing up nicely, and we have good auditory cortex up there. And then if we look at the corrected value, what we mainly see at first is an increase in the R1 values. So what we see, especially in the frontal cortex, is a change in R1 from 0.65 to 0.7, which ends up being a corresponding change in T1 values from about 1550 milliseconds down to 1400 milliseconds, which is a little bit more reasonable of a value. Another apparent change is the slight increase. So here we can see, if we look for it, is this decrease in the B1 also ends up being here in this R1 value, where we see it's much more homogeneous across the frontal cortex. And again, it's important to correct for this, otherwise we might take this difference as being structurally relevant, where in fact it's just an issue with the scanner hardware. So some other work, I mentioned the bipolar study was ongoing with longitudinal studying. So they've switched their imaging approach to do a 2-point R1 fit. So some work that I had accepted for OHBM was to look at B1 correction with block simulations and looking at how that impacted the left-right asymmetry. So I mentioned the B1 field is scanner dependent. So here at this site, we see much more left-right asymmetry. Again, we want to make sure we correct for these hardware issues. Otherwise, we might report this as left-right asymmetries being real in imaging data and putting that onto clinical populations that might not truly exist. So in the uncorrected data versus corrected data, we sort of see a flip. So the last slide I showed, the R1 values increased. Well, here we're seeing a decrease, mainly because these B1 plus values are slightly above 1, but we do see a loss of asymmetry. So they become more symmetric, and to better quantify that, the nice thing about FreeSurfer when you're doing surface mapping is it sort of provides a one-to-one mapping between left to right. So you can calculate the left-right percent difference seen in the signals. So we see, again, very strong asymmetry in the B1 map, where the frontal cortex is really asymmetric at about 15% difference. And this ends up corresponding to almost 10% difference in R1 values in this region. And once we've accounted for this B1 value in the block simulations to calculate the R1 maps, we really see this asymmetry drop off. So that will prevent us from making incorrect statements about asymmetry in the brain when we go to look at longitudinal changes in bipolar disorder. So moving forwards, R1, I mentioned, is also sensitive to iron. It's mostly sensitive to lipid content, but we still have some iron contribution to this. So working towards a more specific myelin marker, the next step would be to use MT imaging. So again, quick refresher, what we're looking at is an RF pulse applied off resonance to selectively excite the bound pool, and then looking at its impact through a decrease in signal in the free water pool. So what this signal might look like, if we're looking at the classic MT SAT approach proposed by Helms in 2008, is for every imaging block, we have a saturation pulse that saturates the bound pool. And this would be the water magnetization if we're tracking it over time. So while we're saturating the bound pool, it's quickly exchanging with the water pool. So we're seeing that magnetization decrease over time. A quick gap between the saturation pulse and our water excitation pulse, which we use to acquire our data with. We see a brief relaxation, and then we see a signal drop as we image the data, and then it recovers over time. So what this proposes to do is to find the percent drop in signal that's achieved by the saturation pulse. And in order to calculate that, we need an MT weighted image, as well as to know the T1, so this relaxation rate, or R1, sorry, and the apparent magnetization. So that would be this sort of the scaling up and down of this line. And if you do that across the brain, you get these nice maps. And on a grayscale, it looks like it's pretty homogeneous. But if you look into more qualitative analysis or change this from a grayscale to something else, you can see that there's some B1 effects in this image. So we might want to correct these images. Again, if we look at a B1 map that we acquired, central brightening with decreases out towards the cortex. If we go uncorrected, we see actually an inverse correlation between the B1 and the MT sat value. And if we want to correct this, there's really two approaches that are being used to account for the B1 plus changes. So one is to correct the excitation and saturation variations after you've combined the two, so that you can use an empirical factor to sort of fit a quadratic function to the variations and then correct that way. The approach that I've taken is to correct each separately. So similar to the R1 map correction, we use the B1 relative field as a multiplier on the excitation flip angles, and then we use sequence simulations to estimate the proportional change that we see from the saturation pulses and correct for that. So we see pretty similar results, at least empirically, between the empirical factor and the simulation-based correction. Sort of work that I showed in ISMRM is how this changes between the more classical approach from VFA. And then one of the benefits of being able to separate the excitation and saturation pulse B1 correction is that we can now use other approaches to generate our M1 or M0 maps and our T1 maps. So again, MP2Rage is a great way to do that. If you look into the literature, they provide a little bit more accurate T1 values. And additionally, it's a little bit faster already than a VFA if you use enough grappa and other things. But there's also a recent sequence by Moussard et al. That used compressed sensing. And by using compressed sensing MP2Rage, we were able to generate reasonably similar MT-SAT maps and P1 maps as the MP2Rage with about 40% less imaging time than the MT-SAT. So that's huge to get really good data quality with nearly half the time compared to the more standard VFA approach. So if we want to look at what that looks like in 25 subjects, again, making the case for this B1 correction being important. You've already seen this B1 map across the cortex. So if we go uncorrected, we really see a strong decrease in these MT-SAT values. I could rescale it and it would probably look somewhat similar. So sort of what's coming up here is the MP. So this is generated from the MP2Rage data. The B1 correction also impacts the T1, but also the M0 values. So that's largely what's driving the decrease, as well as accounting for the B1 effects in the saturation pulse. And then the MT-SAT values look pretty good, good left-right asymmetry, and we're not seeing this decrease in the medial frontal regions that is showing up in the B1 map. So bringing this slide back again, we've fixed MT-SAT to a reasonable extent. Can we exploit properties of cellular structures to get an even more specific marker? So my more recent work has been looking at ihMT, or inhomogeneous magnetization transfer imaging. Again, using the same physical mechanisms as the previous MT imaging. But it gets a little bit more complicated. To take advantage of this, we know that MT is dependent on numerous parameters, including the exchange rate between the pools. So on the far left, we have the free water pool and then the semi-solid pool on the right. So we have exchange between pools, we have relaxation rates, which are, or relaxation times defined by these T1 values, and then saturation rates, which are these R values. We won't get into these equations, but you can basically model this three pool with three sets of differential equations. If you could reach a steady state, you could solve this, but because of safety concerns, we can't do that realistically in vivo. So we have to evaluate these numerically. So how is this used to make a more specific myelin contrast? So we take advantage of this dipolar pool, and myelin contains a large dipolar pool, which essentially is long carbon hydrogen chains. We know from Provodorov theory and from Lee et al. That you can use either a single, like a positive or negative off resonance pulse to partially saturate this dipolar pool. And if you then also acquire an image with dual frequency saturation, so if initially you acquired an MT pulse with, say, 7,000 kilohertz off resonance, or negative 7,000, and then you acquired one where you're dually saturating at both positive and negative 7,000, you get near complete saturation of this dipolar pool. So then the only difference, considering all these other parameters are the same for the tissue with two different images, you're really seeing the difference in the saturation of this term. So this difference in images will report the density of the dipolar pool, which we are suggesting is increasingly specific to myelin, while removing contributions to macromolecular content. So this was initially showed by Varma et al. With some initial phantom work. So we have at the top egg whites, which is ovalbumin, just a protein. We expect good MT from a protein. It's a high macromolecular content. And then in the second row, we have hair conditioner, which actually forms these cool lamellar structures, which would be similar to myelin in a way, high lipid content and phospholipids. And then we have just manganese chloride. So we have T2 values that change, but no macromolecular content. So if we look at the MT, we see good MT for two situations where we have macromolecules, but nothing for these manganese chloride, which is what we expect. And then if we look at the ihMT, so inhomogeneous magnetization transfer ratio, we remove our contributions from proteins and we're left with lipid contributions. So how do we go about doing this? Again, if we look at the more simplistic approach as MT-SAT, we have our saturation pulse. Although in order to acquire a dual, we might split this up into two pulses and have one positive and one negative. And if we do that across the brain, calculate these MT-SAT maps, these are scaled exactly the same, but we see here quite easily that this dual frequency saturation results in a much larger degree of saturation across the brain than just a single offset frequency does. So the same RF energy is being deposited in the brain to get the contrast, but we're seeing this much greater degree of saturation. And then to get our ihMT SAT metric is simply the subtraction of the two images, and we see this value. So it is a little bit noisier, so the increased specificity comes at a cost, but it can be quite useful if we're trying to disentangle contributions from proteins, myelin, and iron content in the brain. So bringing this back, we know that ihMT, how to generate the contrast is we need really high B1 values. So it's really SAR intensive process. And if we look at simulations as B1 decreases, so again, this is the relative B1 value, the single and dual offset MT-SAT maps start to converge. So as we lose our B1 power in the brain, we lose our ability to see any difference between the two situations. So we were able to use, again, the same sort of framework of using sequence simulations to correct for this. So looking in again, a 25 subject average, this B1 map, we see a good degree of asymmetry, again, but seen across all the maps that have been corrected. Primary regions showing up nice and bright. If I put them side by side, you will see a little bit more noise in these maps compared to both the R1 and the MT-SAT maps, but we're seeing very good anatomical contrast here for these ihMT-SAT maps. And again, pretty good correction of this drop that's seen in the medial prefrontal cortex. So quickly going through this. So the next steps for this work that's ongoing is So I showed you a very simplistic sequence case. Realistically, we have a lot of parameters that we can play with in terms of optimizing the sequence. So all of these sort of values are something that we can change on the scanner. So I'm looking at how we can optimize these parameters to improve the SNR and increase resolution of this imaging. Ultimately, so we have a specific myelin marker to investigate cortical microstructure. Try to get through this quickly. So biophysical modeling, ultimately, this is where we want to take this is the next step. So future directions will look to include ihMT to model and calculate the G ratio in the brain. So what is the G ratio? Well, it quantifies the thickness of the myelin sheath. It's a ratio of the axon to the outer myelin sheath radii. And it's going to aid in supporting changes to axon and myelin morphology. So it'll help us disentangle these cases where we're potentially having thicker sheaths on a healthy axon, potentially loss of sheaths, such as might occur in MS, and then in late multiple sclerosis, you might have axonal degeneration. So being able to quantify these will help us disentangle these 4 cases. So some really cool work was done by Sean Dionne's group, utilizing the G ratio mapping technique to show brain development. So this is a 102 days old baby, and then they did MRI mapping. So it's sort of inverse contrast. So a very high value of 1 is basically no myelin at all, and the theoretical value for the brain that we're expecting to achieve based on literature is around 0.7. So this work really shows the development of the brain, which I thought was really cool, from about 3 months old to 7 years of age. Here we're seeing a G ratio value of about 0.8, so there's still some development to go. So it fits in nicely with what I showed, saying that we continue to develop until about 35. And ultimately, what we're trying to do to validate these metrics is look at the marmoset. So at the MNI, we're able to image marmosets, and they provide a very simplistic model for the human cortex, which makes them great to study. They have minimal gyrification, so the brain is much smoother compared to a human brain. But despite that, they have very similar cortical organization and myelination patterns. And just some initial work that I showed at ISMRM last year, we're looking at calculating the myelin volume fraction map and sort of validating the different metrics you can use to quantify this, such as T1 weighted, T1 divided by T2, R1 MT-SAT, and this ihMT-SAT. And then we need to quantify axonal volume fraction with diffusion metrics, ultimately to get a G ratio. So this initial work was just looking in the white matter, but we're hoping to use histology to push this model into the cortical gray matter. So wrapping up, MRI provides a means to study the relationship between brain structure and function, and intracortical signal changes can track aging and disease progression, which makes it very interesting to study. Additionally, these changes that we see can correlate with functional outcomes. So ultimately, the development of specific imaging biomarkers and biophysical models will provide enhanced microstructural information while removing, making it specific and removing hardware-induced variations in the data. So I'd like to thank all my collaborators at McGill, University of Calgary, Lund University, and McMaster. Thank you.
So does anybody have any questions? If not, I had a couple. I was just curious about. I didn't know that verbal memory was affected in bipolar disorder, just because by like the canonical kind of description of bipolar disorder, that doesn't really seem to like fit under. So is there like a strong comorbidity between, I don't know, bipolar disorder and like some forms of dementia, or is there any sort of theories about why verbal memory may be impaired in that disorder?
I'll be honest, I'm not entirely sure. I
Question.
leave that sort of portion to the psychiatrist because they know it a lot better than me. As I've done a little bit of looking into it, but I haven't had the time. But like most cognitive studies, the data is quite messy. And you can usually, almost like nutrition research, find things that say one thing or another. But verbal memory, interestingly, does seem to be one thing that consistently shows up in bipolar disorder. And like you mentioned, it's not completely obvious why.
Yeah, it's often a strong predictor of outcome, but I think Jocelyn is probably the best equipped on this call to talk about that. Just
yeah, if I may, the dementia is twice as common in patients with bipolar disorder over the age of 65 than in general population. And people assume that it's because of the repetition of mood episodes, which then may impair or have some impact on working memory and also executive function. So there are not primary symptoms of bipolar disorder, but probably consequences of the episodes. And on another note, lithium, the main treatment of bipolar disorder, is supposed to reduce this risk. So it may have something to do with myelin. That's why it's what Chris reported is really interesting.
Thank you. Does anybody else have any questions?
Yeah, I have some questions. Chris, great talk. A really comprehensive, sweep of a lot of technical mastery that obviously was needed to conduct, a really broad array of work. So if I look at this from the applications perspective, so you kind of touched on this a bit, where things you might want to do if you're starting your own study. So let's say we do run studies here. What would your recommendation be? You talk about MT versus MP2 RAGE or T1 mapping. Like, where would you start if what you really want to do is examine cortical myelin, and how would you bake that into your study design based on the methods that you showed here? Like, what's ready for prime time based on what you showed here?
Definitely. My experience with the MP2 RAGE has been very good, especially with the compressed sensing one. So it seems like a no-brainer if you can get access to that sequence to use it, because I think we have it running at about 4 minutes to get a very high quality T1 map. So I think I would just use that, and it segments quite well with FreeSurfer. So it doesn't make sense to acquire a different anatomical to me than to just use that, because you're getting a bunch out of that one image. And then because you need that for MT-SAT calculation, I'm not sure if you're getting much more out of MT-SAT than you are out of a T1 image. So I think I would try to go towards the ihMT if you could, once I get it sort of worked out a little bit better. It's almost ready.
I always feel that way. Thanks.
So I would like to ask a question. Chris, very nice presentation, really very impressive.
Thank you.
So I just want maybe that you mention the different ways of normalizing the B1 inhomogeneity. So I saw that you mentioned a paper of yours in MRM 2021, and I will definitely look at that. But before then, you actually presented data from 3 Tesla, and you said, here is the normalized or the corrected, here is the non-corrected. So I'm asking, what are the good ways, or like if you can list the better ways to normalize for B1 inhomogeneity?
Yes, so I think the comparison between 3T and 7T, that was the MP2 RAGE. So Jose Marquez already has code out on his GitHub to do that calculation yourself in MATLAB. It's pretty straightforward. All you have to do is input a B1 map into the function. If anyone's looking to do that, you can contact me. I've also made an additional function to get B1 corrected M0 maps if you're planning to do an MT-SAT calculation. But at least for that, it's pretty straightforward. For correcting MT work, the paper, it's a little bit more complicated. There's a lot of simulations that goes on, and then correlating the simulations to imaging data. But if people are interested in doing that and they're not quite sure what's going on, I can definitely help them out. But it's all on a GitHub that's referenced in the paper, and I think I laid it out fairly straightforward. But definitely T1 mapping is pretty straightforward. It's just how it's calculated, whether you're just multiplying the flip angles by your B1 map. It gets complicated if you're using an inversion, potentially because the inversion pulse may be adiabatic or not. And if it is, then it's likely not impacted by B1. If it's not, then it will be. So there might be, for a specific case, a little bit of playing around to check that.
So it's easier to correct for VFA acquisition of T1?
So VFA is a little bit trickier because there's the issue of incomplete spoiling. There's a couple of papers out on correcting that. So basically VFA assumes that you spoil the transverse magnetization between every readout. So that's quite rapid collection. In reality, we don't end up spoiling the whole magnetization. So there's still some remnant signal from the previous readout. So there is a few papers that have ways to correct that. I haven't implemented them myself, but a few slides ago, I'll see if I can bring it up. It ends up being, anyway, you end up getting lower T1 values or higher T1 values because of that. So if you don't correct for it, you end up seeing those T1 values closer to maybe 1600 milliseconds at 3T, where they are probably more realistically around 1400 milliseconds.
So at the bottom line is that my understanding of all these options, you recommend the MP2 range. Is that right?
Yes, and I think the literature out there, especially in phantom work, is showing that MP2 range is giving more accurate and more reproducible values.
Thank you very much.
You're welcome. Thank you.
So it's noon now, and so I guess wrap this up. And thank you very much. This was a great talk. And so yeah.
Thank you everyone for coming.